Hi, this is the evolution part two notes, evidence for evolution. So prior to this, we talked about some of the individuals that originally started this idea of how things change over time, and we looked at Darwin and his theory and what he used, the finches and the beaks of the finches to um, determine that evolution does happen. And we looked at some of the characteristics that cause evolution, like natural selection and adaptations and the struggle for existence and competition and changes over time. And now we're going to take a look at some of the evidence of evolution. Evidence that things have changed over time. So these are the four major areas: the fossil record, that some species change, some don't, some even become extinct; comparative anatomy, homologous, analogous structures, vestigial structures. We look at embryology, just the beginning of life, looking very similar, and then molecular data, the DNA and protein sequences. So if we look at just history of life on Earth, Earth is about 4.5 billion years old. Life is about 3.8 billion years old, and the order of life as it appears in fossils. We look at organisms. Prokaryotic bacteria are first. Those are the oldest. They went through chemosynthesis and photosynthesis about 2 billion years ago. And then the eukaryotic cells evolved through protists. We can kind of trace our history through life. So if we look at, again, one of the essential questions: what are fossils and how are they formed? Fossils are just any evidence of an organism that lived long ago. Things like parts of an organism, footprints, eggs, seeds, pollen, droppings, complete organisms. And fossil formation occurs when things are buried by sedimentary rock, the sand, the dust, clay, ashes, silt, any kind of sediment. And over time, pressure and time, they form into what's considered a fossil. So, what carries small particles or small rock particles to lakes and seas? Then the dead organisms are buried by layers of sediment and form new rock. And then when we dig down, the older layers are, of course, further down in the sediment and can be dug up. Here's an example of the oldest layers at the bottom, younger layers at the top. And you can kind of look at the bones here in the fossils to determine, which are the oldest and what are the youngest, and what kinds of organisms lived during those time periods. So if we look at the fossil record is incomplete because some evidence about history, but of course it does provide some evidence about the history of the Earth. When we look at the trilobites and the leaf and the caprolite, and these just sort of show a progression from ancient primitive to modern day forms. So if we look at how old is a fossil, right? Fossil record. Again, the history of life on Earth can be partially constructed by dating and comparing the fossils. We can use relative dating or absolute dating. Relative means sort of the approximate age. So if we said, for instance, the relative time period doesn't really give us the exact date, but it gives us a relative kind of period of time where these organisms existed. Absolute dating is the exact age determined by radioactive dating. And so if we look at relative dating again, it's just an estimate. The older layers are on the bottom, the younger layers are on the top. And of course, index fossils are fossils that are well known, existed for a short period of time, and had a wide range of geographic area. But we can determine, based on these layers, that if we know when the volcanic ash from these volcanic eruptions occurred, then in between there we can sort of get a roundabout year of when the organisms actually existed. So if we look at radiometric dating, this is going to help us determine the exact age, the absolute age that determines the exact age of the fossil by measuring the amount of radioactive isotope that remains in a sample. Because a term that you're going to become familiar with, and we'll have a lab on this next class, is the word half-life. A half-life is just the length of time required for half of the atoms to decay. So again, looking at the mineral sample containing radioactive atoms which decay into daughter atoms. So percent of radioactive. So every so many years, the sample is going to be cut in half. So if we start out with 100, when it reaches half-life, half remains. Half of a half then is a fourth. Half of a fourth is an eighth. Half of an eighth is a 16th, and we keep on breaking it down until none is left. So, what is comparative anatomy, and give an example? So, comparative anatomy, we want to compare the anatomy of different organisms. The first term is, again, we're looking at evidence of evolution. So we look at the structure of organisms to tell us a little bit about the history. So the first term is a homologous structure. Homologous means same. Structure develops from the same tissues but different forms from different functions. The same origin, different form, different function. Divergent, the term we use, divergent evolution, where we have things that are similar, different form, different function. Things evolve similarly, but have a different function. This doesn't, for instance, we have a human arm, leg of a cat, the wing of a bird, all have similar bone structures. So the similarity just means that if we have similar structures, then we might have come, or the similarity is due to having a common ancestor at some point in time. So again, looking at divergent evolution, I'm going to move this for a second. Many different organisms evolved from one that had a similar structure. Similar structures. Different organisms, similar structures. Divergent evolution. All right. Similar forms, but then they diverge, they break away from each other to form different species. So what's an analogous structure? Analogous just means different origin, similar function. For instance, human, bird, turtle, elephant. Different form. They use the same function to move. Right? These are different forms, but they all use them to move. So analogous structures like an arm and a leg, same function, but they came from different, they evolved differently. That's also called convergent evolution. They evolved together to have a similar form, but their structures are a little different. Vestigial structures. Structures that were used in the past but not used today. Over time, natural selection eliminates a certain organ that's not useful. So in snakes, pelvic bone or the hip and the femur and claws are present in many snakes. Really useful. Pelvis and back legs in a whale. I don't use them. There's an appendix in a human. Not useful. Used to be useful, now they're not. It's like in a cow. Cows use an appendix to grind up raw plant material. So the theory is that maybe humans at one time needed to grind up raw plant material, or that human stomachs evolved over time to be able to digest everything, so there's no need to have an appendix, and now we're left with a non-functioning appendix. So here's the pelvis and femur of a whale. Not really useful. No function. So what's embryology? This is another embryonic development. The early stages are very similar. Look at the fish, the chicken, the pig, and the human. Early stages are very similar. So this just suggests that maybe at one point we shared a common ancestor. Here's another example. Fish, salamander, tortoise, chicken. We all are animals, might have come from a common ancestor at one point in time. So looking at molecular evidence supports the theory of evolution. All right. DNA, this is what we're talking about. Looking at similar, move this again, similarities between the DNA or protein sequence of different organisms show relationships. Gene sequencing, right? Looking at the letter combinations and the DNA code. Very similar, maybe just a few minor differences. Again, the DNA structure will form proteins. It's the code to make proteins, and if we have the same proteins, we're going to be similar. And so the idea is that, again, that we might have come from a common ancestor. So what we're going to do, we're going to stop here. The rest of these notes we're going to